All right, thank you very much. And it is 8:10 in Cairo.
It's fine, Rob. We are enjoying it. So if you can tolerate the 10-15 minutes of questions,
I can tolerate
we had
another hour or two.
we had
I'm
some
very tolerant.
we had some technical issues with the questions as well, but I think they are open now. So and if not, then we can open the chat. So please feel free to ask questions or even raise hands. I will try my best to see if somebody has raised hand and give you the mic. Anything that can allow us to ask question will be fine. There is Ibrahim. Let me try to give you the mic.
That was awesome. Thank you so much. We would be happy to learn. Then it got cut off, whatever the rest of the question was. So you have to read it to me, Rami, or
okay.
Or let me. I can go to the. Sorry, I can go to the chats. Duh. Okay. So the conclusion, insight. Somebody asked what was on that last slide that I said was important. What's important is you don't really need a wet lab to do great science. You could be a high school student in Cairo, highly motivated, and you just need the gene network URL and access to maybe an AI chatbot like Opus 4.6, and you could produce a paper a day. And I'm not joking. You might not be able to write a paper a day, but you could do all of the analysis required in a day. I can do it in 3 hours. I can take, so I could take Rami's data and all of Malakop's data. I could go every day. I could probably do the equivalent of a new results section for them. There was method sections already done. They already told me what they did. I just took their old data, looked at it, more than a decade later, improved it greatly. And then I have instead of in PLOS pathogens, which is a great paper, I might have it, I don't know, in some other paper, in some other journal. So I think there's a feeling that you can't do science unless you have a lot of funding. That's true in certain projects. If I want to do proteomics for 10,000 animals, I would need a great deal of money. But I don't, because we already have proteomics from the world's best proteomics group at ETH in Zurich. So you can look at that data. There's all kinds of data you can play with. So let's see, what other questions do I see here?
So there seems to be a bug with the Q&A tab. So if you want to put your questions in the chat tabs, everybody.
Okay.
I have a question until everybody gets their thoughts. I have a very simple question, but you started and ended with the same topic, basically, is that data should be shared, open data, and once you publish, you have to put the data out in a shareable way, in a usable way. And we know that many people put papers with data that are very hard to use, the supplementary, like you said, supplementary files, etc. But also you ended with the same note, and you said you were the first to put your data out with the gene networks in 1994 and so on. But I'm sure that you agree with me that not everybody has the same generous vision as yours. Some people like to keep their data until they get like 10, 15 papers out. And others are very suspicious. They're not trying to protect their data out of greed or something, but they are suspicious that someone, what is circulating, some other other competitors or another country and so on. So what's your view in general on openness of data, and how is it just utopia or is there danger,
So in 1971, Rami, I was a classic hippie. I listened to the Grateful Dead. And the last thing in the world I cared about was making money. I wasn't rich. Buying a new toothbrush was a big deal. Even so, but I realized I wasn't going to starve. I lived in America, and eventually I got into biomedical research. And again, I did it out of a love for research and the questions. I didn't do it because I wanted to have an 80-foot yacht that I could sail around Sicily And, that wasn't the goal. So I guess, you have to decide at some point in your life, are you in it for money? In which case, you're probably in the wrong business unless you go into big industry, big pharma, in which case, okay, you're in it for money, go ahead and make a money and then retire when you're 60 or 65 or do whatever. But that wasn't my goal. And I think most people that are doing biomedical research, it should not be their goal. If they want to get rich, there are better ways to do it, believe me. Jump on the AI bandwagon right now. So my attitude has been, I'm going to do what I care. It turns out, Rami, that I'm lucky. Nobody else does what I do, so I have no competition. Nobody's as crazy as me, apparently, to do what I do. So I just do what I do, and I just throw it out there. Now, if it were human data, there would all be all kinds of protection issues. And that's why I generally don't work in human populations, because I don't want to have to dance around the legalities. I don't want to have to go to jail because I provided somebody with data that I shouldn't have, and it got loose on the internet. But mice unfortunately don't have the same legal rights that humans have, so I don't have to worry about it very much.
Yeah, that's an interesting view, but still, well, we can discuss it later. Of course, I think you are a big believer of putting the open data out there, and maybe I agree with you too. I have the same vision, but like everywhere in the world, some people produce the data and put them generously, and others maybe not sharing. So that's the problem, because I think Gene Network gets better and better when everybody can put the data in a shareable way. Anyway, I don't want to take all the time for questions. Now we have a bunch of good questions.
Yeah, I see them. Let
So
me just run down them in
sure.
series.
So
So
one,
Mark George Emil has a question, and that's whether gene editing interventions could potentially extend lifespan. Absolutely. There's almost no doubt that it would be true in general, but the effect sizes are going to depend extremely significantly on the environment. So not only would you have to do the gene editing, Mark George, but then you would have to tell the kid when it was about 4 years old, sorry, you can't have any candy because we edited you not to have candy, or you're too impulsive, so you can't have a fast motorbike. Uh, you'd have to do both at once. I don't think humans are smart enough to do this yet. Um, and I would not trust humans to even genetically engineer a mouse to consistently live longer. Now, we know that some mice in a laboratory environment will live to be 1,200, 1,300 days. That's huge for a mouse. But what do you think that same mouse would do out in the wild in Cairo, fighting off rats and other mice and cats? It might live for 2 days. So it's all about the G by E, and we can control that in mice to some extent, but in humans, we can't. And so if you genetically engineered today for a kid, well, maybe you may. So coming back to this, I think where we need genetic engineering is where we know the environment is going to be radically different. So if we have colonies on Mars in the next 500 years, those people are going to need radically different biology. Gravity is different, so much is different. They probably will have to be more easygoing because they're going to be living in little huts together. So there you might really have to tweak genomes in order to prevent humans from killing each other. There's a funny thing that says that it's amazing that you can put a bunch of great apes in a metal tube and fly them from London to Perth, Australia, or Sydney, and they don't kill each other. They're crammed in one metal tube for like 20 hours. You couldn't do that with gorillas or chimpanzees. You might be able to do it bonobos. That would be a comical that would be a comical flight to imagine a flight of 400 bonobos. But it's all G by E is the answer to that. And then there's another question that looks great from Mohammed. Um,
Mahmoud
I'm
Fahim,
curious.
yes.
I'm curious to hear your thoughts as the founder of Genera, how we could tell. Oh, this is whether more data is inevitably better. And the answer is no. You really have to look at the data. And this is why the QC step is so horribly important, because you can contaminate the data. And we are going through all of our expression data sets, Mahmoud, to look and make sure that we didn't add some bad data. And often we will swap samples. So BXD1 got swapped with BXD2 because they were next door to each other in the plate, or male got turned into a female in our database. So it's a constant struggle to clean it up. I would say in general, though, yeah, in general, more data is better. And I just wish we had more proteomics data. We have a huge amount of transcriptome data. We have almost no proteomics data. I hope that answered that.
Yeah, there are two more questions from Zainab and Ibrahim.
Yeah, I see Zainab's question regarding the ability to just go and analyze data that's out there and publish. Unfortunately, we're limited to the computational resources. Do you know a platform like Kaggle, et cetera, well, not yet. So I do know that there are free subscriptions, obviously, to most of the big large language models, OpenAI, uh, Anthropic, Claude's Genesis. Those are, so you get a free level, but it's not really enough. So I started using Claude about 2.5 years ago for free. And then I said, oh, hell, I'm going to have to spend $20 a month to upgrade to a pro level. And I loved it. Now, $20 a month is not tragic. If you're American, that's like half a tank of gas. So basically any American should almost be able to afford $20 a month. We do for our TV. If you want to get some sports channel, it's way more than $20. It's going to be $100 to get a sports channel. And then after about a year of using it for $20 a month, I got so hooked on it for research that I now spend $130 a month. But that's too much, even for the faculty in our department. So I did go to, well, I basically looked at our department's budget and I said, okay, I'm going to get Anthropic Claude for all faculty if they want it. So now we have a Claude Anthropic account for faculty, which is probably going to cost the department close to 10,000 euros per year. It's, I think, well worth it, because if you're in Cairo or any place without access to cutting edge laboratories. I certainly am not. UTHSC is not MIT, Stanford, or Caltech, just to be absolutely clear. we are essentially a third tier institution. We do some clever stuff, but if you're in that boat, and I would say that the MARC is going to be as strong or stronger than us. We think it's worth $10,000 to spend the money to provide AI to a small group. And our small group is going to be about 20 people are going to be using it. And if it costs us 10,000 euros per year, it's well worth it. So, how do you integrate digital? So Abraham asked,
Ibrahim.
how do you integrate digital neuroimaging data with genetics and epigenetics to distinguish causal effects from correlation? That is a great question, and I'm sorry, Abraham, that I didn't get to it. The answer is large sample sizes. So I showed you data for 57 in the paper, and that's only 4 BXD strains in that paper. Not nearly enough to do any genetics, and not nearly enough to go from correlation to causality. But I mentioned we have 600 plus that we've done. That's for 24 strains. That's the same sample size that Rami and Malik used to study strep. So now any one of these traits, I can map it. So I can say, does it map here? Now that's not mechanistic causality, it's genetic causality. There's a big difference. I can say, I know the Huntington gene is on chromosome 6 in humans and it causes Huntington. But that's a world of difference between saying that and saying, I know the mechanism of Huntington. Still, after 30 years, we don't know the mechanism of Huntington. We know there's a CAG repeat, it does bad things to neurons, but that's not a mechanism. That's just phenomenology. So the hope is you can go from correlation to a specific locus to a specific mechanism. And Abraham, have a look at the Nature paper when it comes out. There's a whole section. The last section in the results says, here's how you go to mechanism. And we kind of sort of succeeded. It could have been better, but it was a good start. And every time I submit a grant application, people who are used to doing experiments one at a time, they'll always say, but Rob, you didn't tell us what the mechanism of aging is. And I go, come on. I wish it were that simple. There are about 100 mechanisms. Let me give you the context. Let me give you the geography of mortality. Then once we have the geography of mortality, you can tell me where the Nile originates. Is there one origin of the Nile, or are there two origins of the Nile? Maybe there are three. But you have to answer that question. You have to answer, where's the origin of the Nile? And we're getting there, but we're not there yet.
Thank you, Rob, for this point, because I think we are all suffering from this reviewer who always, by default, asks you first, correlation is not causation. We know that. We even know it before starting the paper. And we have to say it every time. And then, even if you have some causation, but you need to know the mechanism. I wish I knew. Yeah, you're right. But the reviewers now find it very easy, especially the reviewers who use AI. Now we have a great question from Karim. I hope you can address it, because it goes back to my point about open data.
Yes.
You can see it.
Yeah. So Karim, let me just read it. Shall we add the owner of the data in the authorship of the final manuscript, or just give him or her a big thank you in the acknowledgement section? The latter. So I don't expect anybody all the data in the Nature Neuroscience and the Nature paper is out there, and we hope brilliant people will do things we never even thought of. And if they just say, thanks to this paper that was published then by those authors, we're perfectly happy. If on the other hand, Karim, there's a second level. If you contact me and say, Rob, I need you to do X, Y, and Z because I didn't understand X, Y, and Z, it is my obligation to help you. First, I should say, oh, sorry, I did a lousy job on the metadata. I always think I'm doing a good job, but I did a bad job. So let me clarify that. That doesn't involve co-authorship. But if you say, would you do X, Y, and Z for me, and I spend a week doing it, then yeah, then I do expect to be a co-author. And so here's the problem I have. People will write a brilliant paper using the BXDs. They get it published. They use Gene Network. They never cite Gene Network. And then after they're done with the paper, they send it to me and say, would you put all of these data into Gene Network? And I go, you want me to spend about 2 weeks cleaning up your data and then putting it in, and then you didn't even bother to cite Gene Network, and you didn't bother to put me as a co-author just because you published the paper before you asked me. And that aggravates me. And I've had some very close colleagues of mine do that, I mean, including postdocs. So yeah, so those are my kind of rules.
And that's also a very important point, Rob. I'm sorry that I'm intervening, but it's important because, well, first of all, maybe even you don't want to be cited as an author, because not only because you say they used my data, but maybe you don't trust the final product. So authorship is not always. Some people would really like to be author on everything, but others would say no, I want to be author only if if I can trust the paper. But the other point that you say is exactly typical. Like, look at BLAST, for example. BLAST is like cited a zillion, 100 million times, but still a lot of people use BLAST and they take it for granted and they just put the URL. Same, I think, with Gene Network and other tools. So people who produce tools like Rob, they don't want the credit, but they want to be funded later by saying that these tools are useful. So it's really important to cite them because that's the work they did, you know. So yeah, and I think there is one more. Well, it's kind of a standard question in these webinars, and I think it's worth having your opinion as well. It's by Karim as well. It's he's just saying I have to start this journey being a PhD holder in biochemistry. So what's your advice for someone who wants to take neuroscience?
So I'm interested in. Let me just read Karim's.
Yeah.
I'm interested in neuroscience. If I'm interested, from where exactly I have to start this journey being a PhD holder in biochemistry and in the faculty of pharmacy. You don't need to go back to school, Karim. Don't do that. I have a PhD in physiology. I put electrodes into animal brains for 20 years. And then I said, I'm getting sick of killing animals this way. I'm not really learning what I want to learn, and I wanted to scale better. And I said, okay, I'm going to become a geneticist. And I switched over, and I didn't take any genetics classes. I just read. And now you have the internet, and now you have AI coaches. So you just have to motivate yourself and drive yourself and find interesting questions and learn as you go. I had to learn genomics. I was a physiologist. I had to learn state-of-the-art genomics. It took me 10 years to do that. I started in 92 and I wasn't really good at genomics. I didn't really understand the lay of the land. I didn't understand the geography of genomics until 2002. And then I was really at the forefront. I could talk to anybody about genomics, but that was 10 years of self-teaching, autodidact. So the wonderful thing is once you have a PhD and you have the scientific method stuck in your noggin, then all you have to do is apply that for yourself, by yourself. It's more efficient. And now with AI, so I just 3 years ago, I decided I needed to learn more about philosophy of science because none of us learn philosophy of science. Now I'm old enough, I can say, okay, I'm going to learn philosophy of science. So I can read Heidegger now, no problem. But I couldn't initially because I didn't, it was just a different language. But again, using Claude, I said, what does this mean, Claude? This guy is crazy. He can't write worth a damn. And Claude would patiently explain, how to translate Heidegger into what I understood. So you can almost learn anything at this point. As, obviously not everybody's going to be solving the conundrum of dark matter and dark energy and how the Big Bang started, but you can make a lot of headway on anything, Kareem. So definitely you can get into neuroscience. There's some great books. Gordon Shepherd's Synaptic Organization of the Brain is probably the best single book I would recommend.
Thank you so much, Rob. I think we're approaching the end of the official time for this webinar, and we still have hundreds of questions for you. So I'm sure that you allow me to share your email
sure.
and your contact information. We will put everything on YouTube, even because the image initially wasn't very good for the share. So I will put the PDF one way or another on a drive and share it with the YouTube. I just want to conclude by thanking you really, not just for the scientific material, but for all the insight you have given us. And I can watch the emoticons. That's the new language of reactions on Teams. People are putting hearts and many things all the time. But I want to allow myself to ask you one of the oldest questions ever to conclude our talk, but I'm really going to ask it very simple and provocative. With all this advance in neuroscience, biology, but also in AI, will we reach immortality one way or another? Will future humans, well, if President Trump allow us, will future humans be able to live forever?
Obviously, I can't say no. I would be pretentious. I can say, let me put it in my terms just for me. What happens in my brain, I consider to be wonderfully simple. I'm a couple of million years away from a chimpanzee or a bonobo. So it's not as if what's special about any human really is our linguistic abilities, our abilities to model ourselves. That is a very thin layer on top of a primitive animal brain that's not much different from a chimp or a mouse for that matter, or even a snake, alas. So there's a continuum, and we humans have been extremely fortunate over the last 100,000 years since humans moved from Kenya through to Morocco, through to Egypt, through to Palestine, through to Turkey, through to Europe, to acquire this very, very rich, complex culture, including most of all language and communication. And so I consider if you say, if I wanted to be immortal, what do I want to be immortal? Do I want my body to be immortal? Sure, I wouldn't mind being 23 years old again. But what I really consider me is whatever the heck this is that's going on in my noggin right now, consciousness, self-consciousness. That I consider to be, if it's 1 terabyte of data, that's amazing. I should be able to fit on this phone. This phone has 1 terabyte of RAM, and it's got a humongous processor, an A13. There's enough to run Rob on this. It's just we don't have the algorithm to run Rob yet. So, yeah, I'll be happy if my great grandkids can carry me around in their pocket. It would be on a much smaller phone. It'd probably be a size of a fingernail by then.
Yeah, we all hope. This was the oldest question. I think this was the motivation for all the discoveries of medicines and drugs. What do they call it? The magic potion, or so. I hope we find one solution. I allowed
Yeah.
myself also to put your second or third slide here as a finale because it's amazing, and we should all stick with this smart data and with the fair sharing of data. So again, please allow me, if you have been here from the beginning or near the end, I want to thank Professor Rob Williams very much. He's the chair of genetics, genomics, and informatics, in fact, at College of Medicine at the University of Tennessee Health Science in Memphis. And I want to especially thank you for highlighting my paper from 20 years ago. But I want also, again, I want to correct the record. I was the second author on that paper, which was the PhD work of Professor Nurten Abdeltaweb, who many of you know. So thank you so much for reviving our old paper, and maybe somebody will be interested in analyzing it again. Again, we don't want the time to end, but some people have to go to sleep, and others may have to work in other countries. So thank you so much, Professor Rob Williams, and everybody for being here.
And feel if there's one thing I like to, one type of email I love to respond to, it's an email having to do with using gene networks. So have at it. All right,
yeah,
bye everybody.
We all encourage you to use gene networks, explore it, and ask Dr. Rob Williams questions. Thank you so much, and we will conclude now the webinar, and you will find the recording on the chat later, but we will also post it on YouTube. Thank you so much, and have a good rest of the day. Have a good night, those who are in our time zone.